Good afternoon, everyone. I hope you're doing well. I miss seeing you all, and I hope this transition has become easier for you as the days have progressed. I wanted to make another PowerPoint for this lab and try to clarify some things about the lab as we move forward. So this week is week 12, the week of March 23rd, and this marks the first of 3 labs that will end off this new kind of hybrid online lab course that this has become. And we have a few more assignments to do. There are actually 3 assignments related with this particular lab, 3 of which are in, all of which, are in your lab manual. And then there are also 2 online modules that are not available yet, but will become available week 13 and week 14. And then that will be the end of the lab. So if you have any questions about previous lab grades or anything like that, make sure you do check your physical attended lab grades and make sure they're all in order. If not, please send me an email. All right, so this week's lab, we would have gone outside to collect data, but hopefully you're staying indoors, staying healthy and safe. I wanted to give you a little collection of all the different pets I've been seeing on my different Zoom meetings. Up here you can't really see, but my best friend's cat, Chubbs. Here's his head. I don't know why it cut in between. My friend Shelly's cat, Mabel, this kitty here. These are my cats, Eddie and Magellan. So hopefully you've got a furry friend at home to keep you company. And it's kind of ironic, this lab is about spatial relationships, and hopefully you're practicing good spatial distancing if you're not feeling well. All right, so we're moving on to a section about ecology. So that is simply the study of basically how organisms interact with their ecosystem. So eco, standing for the earth, and ecosystems essentially. And this includes a lot of biotic as well as abiotic factors. Biotic simply means a living factor. So biology is the study of living things. Therefore, biotic factors are living. And that can be, we have a lot of examples on the next slide, but there are also abiotic factors such as temperature, weather, things like that, that are not alive, that can influence an organism and where it lives. So, for example, some living components, the amount of food, competition with others, disease, parasites, plants, other animals. This is particularly talking about for this alligator here, what might affect it. Some abiotic components that might affect the alligator are temperature of the air or water. So they are ectothermic, they can only live in the southeast. The climate, sunlight, the amount of oxygen, and even pH and salinity. So things can be arranged in different ways in 3 main patterns of dispersion. So this dispersion refers to how organisms within a population orient themselves within their groups. So they can either be clumped. So this could be they're clumped around a source of nutrients. It could be even, which is what is recommended during this time, that we keep a 6-foot distance from each other, or random in that their location is just not related to anything. So here are a few examples of those different patterns. Schools of fish clumped together for protection. This is a random pattern of trees dispersed and growing in different locations. This is an even dispersion pattern of penguins, which apparently evenly distribute amongst their colonies to distribute heat. So how do we quantify these patterns of dispersion statistically? Well, we're actually going to do 3 different tests, the first of which is called the Clark-Evans nearest neighbor method. So it will actually take measurements between an individual and its nearest neighbor of the same species in order to try to find out what dispersion pattern it displays. So it's going to come up with an R value, and that's going to be tested statistically and using a t-test to see if the arrangement is either one of these 3. Okay. And at the very end of this, there is a chart for determining the statistical significance of each test. So don't worry about that. You can also send me questions as well. The second test is called the variance mean ratio method. And this looks at the dispersion pattern by counting the number of individuals in a randomly sampled quadrat. So this uses different symbols as well. There is a lot of math involved in this lab, but this is the notation for this specific test. In your lab manual, it goes into more detail as to what these figures mean. And it has spots where you can record each one and explains, like, for example, what is s squared divided by x bar. So that is the variance to mean ratio. So all of these explanations for these symbols are given in your lab book. And for this second test, you can also use a t-test to determine the significance. For the third test, we actually use a chi-squared test, and it's a bit different than the other 2. So we want to see about spatial arrangement once again, and if it's influenced by another species. So we have species A and species B, and we're going to basically see where both of them are present, which is this case W, and that will, those results need to be added across to here, W plus X, where species B is present, but species A is absent, X, where species A is present, species B is absent, is Y. So going down, going across, where both species are absent is Z, down, across, and then add all of these together to get N. And you will determine if it's significant using a chi-square test, comparing it to this number that you get. It's a bit different from a usual chi-square test and is a bit confusing, but hopefully you will be able to get the hang of it. So there are notes in your lab book. The data analysis notes are on these pages 187 to 191, and there are notes as well to fill out and calculations to perform. Okay, so these hypotheses are actually given to you in this PowerPoint as well as the lab. So it wants you to give a statistical conclusion as well as a biological conclusion. So mathematical conclusion, and basically what does that mean biologically? Yeah. This notation is a bit confusing. If you have problems with this in particular, please let me know. For each test, this will be different. Remember, for the first 2, it is a t-test, and the next one, it is a chi-square value. So making a statistical conclusion, remember that in biology, we like to use the p-value of 0.05, and you use the associated degrees of freedom in order to choose the appropriate level of comparison. So for us, that would be down here. 0.05, if we were to have 40 observations, we would compare that to 2.021, our chi-square alpha value. Oh, sorry, t distribution value, alpha value. My apologies. And as this says, we are pretty familiar with this. If the p-value is less than or equal to 0.5, we reject the null and accept the alternative. If the p-value is greater than 0.5, we can fail to reject the null. Hopefully that's correct, and you guys have that correct in your head by now. Okay. If not, please email me or refer to the lab manual. It's got a lot of great resources for this lab. Here is a chi-square distribution table as well. Once again, we focus at the 0.05 level, but these values expected will be given to you. And then you have to compare what you get in your test. Okay? This is what I mean by the hypotheses are given to you, what you would expect from each type. This is supposed to be A0, A and arrow. Sorry about that. This for each test is explaining what you should have for each degrees of freedom, what you should have for each critical T value, your degrees of freedom are. So it gives you a lot of good information, but you actually have to do an online simulation where if we were doing this lab face to face, we would have gone out to the field and thrown hula hoops and looked inside the hula hoops for 2 different species of plants. So this is actually a bit easier in that you don't have to go outside, it's not hot, but sadly you don't get to see some plants or throw hula hoops or go outdoors for a lab, which I think is always fun. I'm going to walk you through the online simulation. So if you haven't gone to BioViewer, it's a great resource for your course, especially if you're in the lecture. There's a lot of material on here that you can use. And as you can see, there are things like lab techniques as well, microscope slides for those of you who are interested in looking at more of those. Population ecology, which is what we're focusing on today. So to get here, you need to go, you can either search for BioViewer or type in bsc.ufl.edu and then find BioViewer from there. And it should be fairly easy to find us. But you will probably run into problems with Flash and Adobe plugins. So make sure you allow that or reinstall the latest version. So you're able to see everything on BioViewer. The password to access this is anatomy. So once you get to the website, enter the password. It says welcome student, and you have a lot of modules you can pick from. So for this week's lab, we are choosing population ecology. For test 1, it brings up a bunch of different things called schemas, which are just different tests and simulations we can run. So for the first one, we want to do Clark Evans nearest neighbor method test. So here it's noted by a star. We just want to do the nearest neighbor schema. We select that. And then we get all these different things that pop up, these 3 different windows here. So don't worry too much about that. Excuse me. The schema is nearest neighbor. Excuse me, once again. From here, please click on quadrants. Once you do that, this will come up. So there are 3 different quadrants as were represented by the 3 different columns. If you click on the plus symbol, it will zoom into that particular quadrant. So all of these green dots within here are randomly generated individuals within this quadrant or population. So you can add a point by hovering over a dot, clicking on it, and extending to another point, and recording the distance in between those neighbors. So this could be one observation from here to here, and this as well from here to here, because they are both nearest neighbors with each other. They will be the same distance if you click directly on each circle. And that counts as 2 observations you can record. You add a point, you click on that one there, extend to the next point and click, and see I didn't quite get it on the nose, but the distance is 180. And I believe that's centimeters, but I will have to get back with you on that so we can calculate area. Then you press add point again, and you add a point there. So that then becomes 0.2, and you can measure the distance back to 0.1 if you'd like. And that's 2 distance measurements. That's 178, whereas the first one was 180. It wasn't quite on the dot, right? So you want to repeat that until you've gotten 50 measurements recorded. So that's the data you'll record down, and you'll record that on page 183. I'm not going to have, I'm sorry, not 183. This is 186. The record is this is in table 10-1. For the last 2 tests, you're going to want to record your data on page 187, and you will be recording the number of individuals as well as presence absence data. Okay, so for this one, there are 4 different schema to pick from. They each just have a different spatial distribution for each population. So, gator pride, we've got orange and blue beetles here. Mean variance schema 1. So whichever one you pick, there are 4 different ones. Just make sure you're right which one you do. And we want to click quadrants again. So once again, we're going to zoom in on this top right quadrant by clicking on the zoom plus arrow. And we get a really big plot where we can toss hoops. So we're going to toss hoops 25 times, and it simply throws out a circle onto your quadrant. So once you throw that out, you want to record data for both tests by saying which species you have. So for example, let's call this species A. We've got 2 species A in our first hoop. By default, the orange dots are species B. We have 0 species B, 2 species A. That means species A is present, species B is absent. We're recording the data for both of these at the same time. You want to toss the hoop 24 more times after that and record the data for those as well. So once again, this is the table that you can use to determine the significance of your test, and this will be available as well. So please email me if you have any questions or concerns about the rest of the course or how it's going to go. This is the last lab that will be from the manual. The other 2 labs, the last 2 weeks, are going to be online modules, but they are not yet open. So you have until midnight on Sunday to complete these assignments. I believe it's the 29th at 11:59 a.m. So once again, let me know if you have any questions. I hope you're doing well, and have a good day. I don't know. I miss you guys. So have a good one. and talk to you next week.